It's a great pleasure to be here, and very happy to be here in Curie, and really honored to be asked to be the keynote speaker of this particular symposium colloquium meeting. So thank you for having me. There are other people in this audience who could speak as well, if not better than I, about X inactivation, but I'll do my best now. So I think hopefully most of you know what the process of X inactivation is about, how to balance between the sexes in mammals by shutting down one of the two X chromosomes in females. So it's important because the X is big. It carries more than 1,000 genes in humans and mice. And compared to the Y, which only has very few genes, and in fact very few genes that actually really matter for guys, there is a bit of an issue with dosage. So having two active X's in females versus males would just not be, or is not viable. And over evolution, as the sex chromosomes evolved in this way, processes had to be developed or evolved to deal with this imbalance. And so indeed in mammals, it's thought that the common way is to shut down one of the two X's, and that this happens randomly most of the time in most mammals, in eutherians at least, where either the paternal or the maternal X is silenced. And this was first put forward as a proposal for how this dosage compensation could happen between the sexes by Mary Lyon back in 1961, simply by looking at X-linked coat color genes and noting that in females that were heterozygous for X-linked coat color genes, she noted these patches of different color in the fur that were never noted in males, obviously. Well, not obviously, but that was the proposal, that one of the two copies must be shut down in some cells, and the other copy must be shut down in other cells. So this was actually very visionary of her. Before she actually came up with this hypothesis, it had already been shown that female cells have a heteropyknotic bar body, and I'll come to that in a minute. But in any case, her proposal was that indeed most of the genes, all of the genes, in fact it's not all, as you'll see, are shut down on one of the two X's. And then once this happens, this state is propagated stably through cell divisions, through some memory mechanism, which we then and now call epigenetic, to make sure that this state is stably propagated and is present in all somatic cells. So we end up with these clonal populations of cells expressing either X. And this has the remarkable characteristic to it, which means that there's phenotypic variation at the cellular level within the same individual in females. Of course, this doesn't happen in males, which means that for X-linked mutations, the effect is total. So indeed, for X-linked genes that are mutated and are linked to disease in females, one often either doesn't see a phenotype or symptoms, or it's only partial due to this mosaicism. And so this process is indeed found not only in mice, but also in cats and coat color. And there are X-linked genes that also affect sweat glands, for example, in humans. So there are many, many examples now of this mosaicism that's due to X chromosome inactivation. And as I mentioned, it was actually already a process that had been detected, at least, at the cellular level, the cytological level, by Barr, a cytogeneticist, in the 1940s, who noted that in cat neurons, there was this heterochromatic body that was only present in females and not in males. So it does mean that X inactivation is a beautiful model for studying epigenetic control, but also for studying heterochromatin, how this kind of heterochromatin is set up during development, how it's maintained, and how it can go wrong in certain diseases such as cancer. So in a very simplistic way, I'm showing you what many, many decades of work from many groups, many people have come to sort of propose, which is that X inactivation is initiated through a unique region of the X chromosome that was mapped genetically both in humans and mouse as the X inactivation center. And from within this X inactivation center, which is rather large, and I'm not going to talk to you about the organization and the regulatory landscape of the X inactivation center, that will be for other times, but basically from this X inactivation center, a unique non-coding RNA is expressed, Xist. And Xist is what I'm going to be talking to you mainly about today. So Xist RNA is a multitasking molecule, as you're going to hear. It coats the chromosome in cis, and this is an RNA fish to show you this. It leads to gene silencing. It leads to chromatin changes, and it leads to chromosome reorganization. It's 17,000 to 19,000 nucleotides long, so it's really a very long RNA. It's spliced, it's polyadenylated. It looks like a messenger RNA, but it's not. It doesn't code for a protein, and it's retained in the nucleus. So how does it do what it does? So that's what I'm going to be mainly talking about today. So Xist is upregulated early on in development, coats the chromosome in cis, and leads to X inactivation. But once X inactivation is established, we now understand that, in fact, the inactive state can be maintained pretty much independently of Xist. Although, as you will hear at the end of my talk, removing Xist can have some effects. But overall, the inactive state is pretty well locked in. And that's due to a number of chromatin changes that are progressively laid down, including polycomb-associated changes, but also DNA methylation, which very efficiently locks down the inactive state of X-linked genes. We also know that the inactive X changes its organization at the chromosome level. Also, presumably or probably at the nuclear level. It seems to be even more stuck to the nuclear envelope when it's inactive. It also changes its replication timing. So there are many different changes that happen. We don't know how much each of them really matters for the maintenance of the inactive state, but they all seem to be associated with it. And here I just want to highlight one level of change that happens that I'm going to come back to very briefly later on, which is that the overall organization of the inactive X is very different to that of the active X. And this is using both fish approaches, DNA fish, to look at how the X is spread in the nucleus, but also chromosome conformation capture technologies that allow you to capture the molecular organization of a chromosome. And this is just to show you the active X is organized into what was mentioned just in the previous talk, topologically associating domains, and the inactive X seems to globally lose its topologically associating domains and is organized rather into two huge megadomains, the function of which are still unclear. So how does X inactivation actually begin? And that's where I'm going to begin my story. So here I'm showing you a very simplistic cartoon of how we think this process starts. And I should say that the studies that people do to look at the initiation of X inactivation involve both embryos, but also embryonic stem cells. And much of what I'm going to tell you today is based on studies in embryonic stem cells, but looking in embryos actually has confirmed much of what's seen in ES cells. So here I'm just showing you a cartoon of a happily transcribing gene on the X chromosome. It's active. It's associated with active chromatin marks such as histone acetylation, HVK4 di and trimethylation. There are transcription factors there. So this now we know, not just hypothetically, but also because people have done chip sequencing. So we have a pretty good idea of how the active X looks before X inactivation happens, and how the active X looks even in the presence of the inactive X in the same nucleoplasm. So what happens is, early on in development, Xist is upregulated, and the chromosome that it's upregulated from becomes coated by Xist. And this leads to very rapid silencing of genes and to loss of active marks, to loss of POL2, of transcription factors. And so much of this analysis was originally done just using immunofluorescence and RNA fish. That's all we had available. But more recently, people have actually used much more sophisticated techniques such as RNA sequencing, chip, etc. And so now we can actually tell you much more about what happens during these early stages, and I'll be telling you more in a minute. But once this has been established, this silencing of genes and these early changes in chromatin states, we then see some other changes, chromatin marks that come in that seem to be associated with this maintenance, and these include the polycomb-associated chromatin changes. So PLC2 is one of the polycomb repressive complexes, and it brings in H3K27 trimethylation. PLC1 is the other polycomb complex, and it brings in H2A ubiquitination. We also see histone variants coming in, and we see other factors, including ATRX, which in fact is an X-linked gene itself. The protein seems to associate with the inactive X in some cases. SMCHD1, which is a factor that seems to be involved in changing the structure and organization of the X, etc. So there are many, many factors that seem to come in progressively and lock in the inactive state, as I mentioned to you. But some genes somehow avoid this whole process. Some genes can escape from X inactivation always in a constitutive way. They seem to be able to be expressed from the inactive X. As you'll see later on in my talk, I hope, these genes are still slightly susceptible to Xist's effects, but nevertheless, they seem to be pretty well expressed from the inactive X chromosome. So how do they do that? We don't know. There are many hypotheses. Is it through specific transcription factors that seem to drive on these genes despite what Xist is doing? Is it due to insulator elements that somehow protect them from the formation of heterochromatin? Is it some kind of special chromatin state? So this is one of the things that fascinates us and many others as well in the field. In addition to these constitutive escapees, we also have some genes that get silenced and then come back on again. And these we call facultative escapees, and they tend to be variable in different tissues, but even between individuals. A gene that shows facultative escape is not always in an escape state and can vary from one individual to another and from one cell type to another. And this is something that also fascinates us. Escapees are often involved actually in chromatin-associated functions, suggesting that they could have global effects when they escape. So one of the constitutive escapees is JARED1C, another one is UTX, and both of these are histone demethylases. And others, ATRX I mentioned to you, is a chromatin remodeler. MECP2 was a surprise to us. It's an X-linked gene involved in Rett syndrome and a DNA methyl binding protein. It doesn't escape in most tissues, but what we found in a paper published several years ago in the lab was that actually MECP2 does escape in some cell types in the brain. And so this is a typical example of a facultative escapee, and in fact many genes around it also show this escape. So the questions that my lab has been interested in, and I decided today that I would actually show you quite a lot of just very recently published data, some new data that we have in the lab, but that opens up questions, and it would be great to have feedback. So we're interested in how Xist does its job, and how, when Xist goes wrong, anything goes wrong, whether gene dosage could be important in development and disease. So the questions we were asking ourselves, and we're not the only ones, is how is gene silencing actually achieved? What are the partners of Xist RNA that allow it to do this, and what do they do? And is chromatin actually involved in this process, at least in initiation, or is it just in maintenance? And this is an open question in many fields. You know, how important is chromatin for the actual process of either transcription or silencing? And for X inactivation, we feel that this was an unanswered process, an unanswered question. So I just want to start by telling you that Xist does matter. So this was shown by actually many labs in the past. I forgot to mention them, but Neil Brockdorf, Rudolf Janisch had shown that Xist is key for X inactivation using ES cells. If you knock out Xist, you can't silence the chromosome in cis. But it wasn't actually clear at what level this was really working. And what Maud Borenstein in my lab did several years ago was use single-cell RNA sequencing in embryos to really look at what the impact is of losing or not having Xist. So I'm not going to go into the details of the early events in mouse embryos. I don't want to confuse you. Suffice it to say that in mouse, the initial form of X inactivation is always paternal. It's always the paternal X that gets silenced. Later on in somatic tissues, it's random. It's either paternal or maternal. So what we did was actually knock out or take a knockout mouse of Xist and cross it to a wild-type female, so a male and a female. And these two strains are highly polymorphic, which meant that we could really distinguish what was happening on each X chromosome, and therefore look at what the immediate effects were when you don't have Xist during X inactivation. And here on the left, what we're showing you are different stages of development. So this is the 4-cell stage, the 8-cell stage, the 16-cell stage, 32-cell stage, and then the blastocyst. And we already knew from work in my lab and others that Xist begins to be expressed at the 2 to 4-cell stage. It coats the chromosome in cis, and it leads to gene silencing using RNA fish. We could see that. And by RNA-seq, and this is many genes, all the genes that we could analyze at least on the X chromosome, and by measuring the allelic ratios between the two alleles, the Cas and the black 6 alleles, we could look at how X-linked gene silencing happens in a wild-type situation. And so here, this is the color code, just by looking at the allelic ratio between paternal and maternal using the single nucleotide polymorphisms from this cross in the different genes. When you're biallelic, you're basically white or gray, and when you become silenced and monoallelic, if you become silenced on the paternal allele, you will progressively become red. So basically, what you can see here is that the genes on both X chromosomes are being expressed biallelically, and then progressively from the 4-cell stage through to the blastocyst, you can see progressively less and less paternal X-linked gene expression, only maternal X-linked gene expression, which is red. And so this is what we'd expected. It was nice to confirm it using RNA sequencing, and as I said, this is single-cell RNA sequencing in early embryos. But what we then looked at was what happens if you look in the knockout situation, and that's shown here, and I think this speaks for itself. Basically, you really don't see any X-linked gene silencing. You stay biallelic, you see very little maternal-specific X-linked gene expression. So what Maud was able to show is that in the absence of Xist, you really do not get X inactivation, you don't get dosage compensation, and it was already known that you get early lethality. And here I'm showing you actually the results from Rudolf Janisch's lab many years ago, published in 1997, where they showed that in this particular knockout, if you get rid of paternal Xist, you see early lethality. Even at the 6.5-day stage, you can see severe defects. And when Maud looked at what goes wrong in these embryos when X inactivation doesn't happen, basically a lot. Both in extraembryonic lineages and in embryonic lineages, we think that things go badly wrong. So basically, if you don't get X inactivation, you're dead. And Xist is the key molecule that triggers this process. So how does it do it? What are the partners of Xist that allow this to happen? This sounds like a simple question, but it's taken the field over 20 years to even begin to answer. Don't ask me why. Maybe we were just not very competent, or maybe the technologies were just not up to scratch. What happened, though, was a few years ago, as you can see, many labs actually managed to start to pull down the proteins that link to this non-coding RNA. And so up here, I'm showing you Xist, and I'm showing you here the different regions of Xist that we knew were probably important just because they were conserved. When you look between mouse and human and other mammals, you could see that these were the most conserved regions. And they correspond to these repeats, which are unique to Xist, but repeated within Xist. One of them was the A-repeat, the most conserved part of Xist. And this part of Xist, we already knew from Anton Wutz and Rudolf Janisch, is absolutely essential for gene silencing. If you knock out the A-repeat region of Xist, you can no longer get gene silencing, even though the RNA can still coat the chromosome. But there were these other regions that we also knew were conserved, and for which it wasn't clear what the functions were. Some of them seemed to be important for recruiting polycomb, others for the localization of Xist to the chromosome. Anyway, so there were many regions of Xist that potentially had a role, but as I said, several groups went in and actually tried to pull down the factors that associate with Xist RNA. And in fact, we collaborated with one group. This is the study shown here, Howard Chang, who used a pull-down approach called CHIRP to actually identify the factors associating with Xist in ES cells, with and without the A-repeat in Xist, and also in differentiated cells. And what he found was that actually you have over 80 proteins that seem to be quite specific to Xist, and only a handful of those proteins actually seem to be specific to the A-repeat, because he compared it in A-repeat positive and negative cells. And several other studies used similar but slightly different approaches, using different cross-linking techniques, etc. And there were two groups who actually used a genetic screen approach, the group of Anton Wutz and the group of Neil Brockdorf. And the nice thing was that from all these studies, there was actually a general consensus about which proteins are involved, or which proteins bind Xist and which proteins are involved in gene silencing. This is shown here. And it seems that the proteins that probably are involved in the gene silencing phenomenon are mainly SPEN, and this is the protein I'm going to be talking to you about in a minute. And SPEN came down in all of these different screens. So basically, we know that this protein is probably one of the key proteins involved in X inactivation. So this was a remarkable year for X inactivation, because we'd been waiting all this time to try and understand how Xist worked since it was discovered in 1990. And it was also remarkable because we all agreed for once, which doesn't often happen. So before actually telling you more about SPEN, I just want to highlight another study that came out more recently, because these studies actually all focused on either full-length Xist or the A-repeat mutant version of Xist. And in fact, Alina Shkromatova, who I hope is here she is, yes, good, decided to go for Xist and different parts of Xist using an approach that just came out very recently. The paper was published, so that she calls inkprint, or was named inkprint, which allows you to identify and measure RNA-protein interactions in a rather, I would say, straightforward and very efficient and much, much cheaper way than many of the other approaches that had been used in those previous papers I showed to you. So I won't go through the details of the approach, but basically it's to tag different parts of your RNA of interest. In the case of Xist, it was different parts of Xist. And then to co-transfect with libraries of proteins fused to this MS2 coat protein tag that the different RNAs of interest were linked up to. So it's an MS2 based approach. It looks simple. It actually is simple. It's very efficient, as you'll see in a minute. What was less simple was actually publishing the paper. And I do just want to say that this was a study which, whenever Alina would present it or I would say something about it, everyone would be so excited about. But it was really a struggle to get it published. And I wonder why people can be so refractory to new technologies that actually make one's life easier. But anyway, that's for the reviewers to understand. So here I just want to show you how the impressive amount of information that came out of this study. So basically what Antoine Grandorge did in Alina's lab, and Ines Pinero from my lab. This was a collaboration between our two labs, but it was really very much Alina's project or program. So basically what they did was take either the A-repeat region or the F-repeat region here, or the C-repeat region, and could have taken any other region, but this was already a lot more than anyone had ever done, and looked at what protein partners were being pulled out. And the nice thing was that with the A-repeat region, they managed to pull out many of the factors that we already knew about, including SPEN, but also some of these RNA modifying factors that I'll come to in a second. So these were pulled out using the A-repeat region. And then it turns out that the F and the C repeat regions pull out some overlapping factors, which I guess would be expected because these regions are very close together. And maybe when the A repeat region factors are being pulled down, they do actually link up to both regions. But several that are actually unique to each of these different regions. And these are still very much open to exploration. So in fact, this is an invitation, I guess, to the community to explore every one of these factors, because there's a lot of work to be done, and to really try and pin down what these different factors are doing in Xist-mediated functions. We already knew that this region, the C-repeat region, was important for Xist coating. And in fact, thanks to Alina's study, indeed she pulled out HNRMPU, which had already been proposed to be involved in this, but many, many other factors. So these could be the factors that help Xist coat the chromosome in cis. Anyway, so that's just a little plug, but also an invitation to get in touch with Alina if you'd like to use the technology, but also if there are factors that interest people, you should feel free to go for it. So I'm going to talk to you now about SPEN. Here, first of all, is just a little cartoon of the way people were thinking. So, of course, we pulled out, or the various groups pulled out all these factors. It was very exciting. And then there was the realization for every one of these factors, this is a lot of work to actually sort out. So in my group, we decided that probably the best factor to start with was the factor that had come out in all different screens, and that was SPEN. And here, it was hypothesized that SPEN binds directly to the A-repeat region, which had been shown indeed, and then somehow interferes with transcription and maybe also with chromatin factors. I just want to highlight, though, that there are many other factors that could be helping in this process, and in particular RBM15 and RBM15B. And these factors, WTAP and METL3, had actually also come out as potential partners of the A-repeat region. And these factors are involved in RNA methylation and RNA modification. And so there are studies that have shown that these could indeed influence X inactivation. But it's still not clear exactly how all of this fits together. So anyway, we decided to focus on SPEN because it seemed to be, as I said, the common factor. It turns out that SPEN is one of these huge proteins that harbors many RNA recognition motifs, which is as you would expect. And it does have a conserved transcriptionally repressive SPOC domain, so SPOC stands for SPEN paralog and ortholog C-terminal domain. There are many domains. It's a big protein. It's been involved in many processes. In plants, for example, it's been involved in splicing. We also knew that it was important in Drosophila. In Drosophila, it's called split ends. And actually, the lab just above me when I was here at the Curie Institute, Alison Bardin, or Alison Bardin, to say it in English, or American, she was actually working on split ends, looking at what its role is in Drosophila. So it does have many different roles. In mammals, it had already been connected to nuclear receptor pathways that are hormone responsive, and people had already looked at what SPOC might interact with and found that it does seem to associate with some of these ubiquitous transcriptional co-repressors, and I'll come back to that in a minute. Now, what exactly does it do in X inactivation? So in the previous studies, most of the tests that had been done to validate had just been knockdowns that just looked at de-repression of some genes, and a lot of this had been done also in haploid cells in the case of Anton Wutz's study, or in transgenic models in the case of Brockdorf's study. So we decided to actually look at this using a slightly different approach that allowed us to look at acute depletion of SPEN using a Degron approach. And before I get to that, I have to just introduce our cell system so that you understand what we're doing. So I mentioned to you already that we use these highly polymorphic mouse crosses to distinguish between the two X chromosomes. This is essential because both the active and inactive X are in the same cell, so we need to be able to distinguish. And in order to be sure that we only get inactivation of one X and not of both, or not of either X, because then if it's random X inactivation in a population, it's almost impossible to capture what's going on because both X's are active somewhere in the population. We use an inducible Xist gene whereby TET operators had been knocked in upstream of Xist, and we could therefore induce Xist using dogs. So this was a cell line that was derived from a mouse cross like this, with the inducible Xist in this mouse. So this was actually generated by Anton Wutz. And so we made an embryonic stem cell derived from this. And this was with Edda Schultz and Christelle Picard in my lab. So Edda was a postdoc at the time. She published a paper showing how important X inactivation is for differentiation, in fact. And if you don't induce X inactivation, you slow down differentiation of ES cells. But she characterized this cell. And what we were able to see is that when you induce Xist, you get very rapid gene silencing. And in fact, and so this is by RNA fish. And this is quantifying it by RNA fish, but we also did RNA-seq. And basically, gene silencing happens with very similar kinetics, in fact, to what we see in the embryo. So that was reassuring. We also see there's a very rapid loss of euchromatic marks using IF, and so that was also reassuring. And there's a gain of polycomb, which is the earlier heterochromatic marks, but not of DNA methylation, which is probably as expected because here we were only in undifferentiated ES cells. So this was a system that allowed us to decouple the initiation of X inactivation from differentiation events, because normally X inactivation only happens when you differentiate mouse embryonic stem cells. Here we could induce X inactivation in undifferentiated cells. So it was a much cleaner system for us to work with because there was less heterogeneity, and we could direct which chromosome was going to be silenced. So using this cell line, François Dossin, a student in my lab, and in fact, he started this work when he was just a master's student, and even within the first few months, he managed to generate some of the cell lines I'm going to show you. So he's remarkably talented. And what he did was to use an auxin-degron system. He knocked in the degron tag into SPEN itself, into our protein of interest. So this is the AID tag. He also put this tier 1 locus into ubiquitously expressed locus. And so these were tools that were actually kindly donated to us by Alfej Nora, a previous student in my lab, also very talented, and Benoit Bruno. And so what François did was knock in the tag at the endogenous SPEN gene. He knocked in the AID tag along with a halo tag or GFP, so we could visualize it at the same time. And using this, what you can do is you can induce Xist by DOX, and you can induce SPEN degradation by adding auxin. The addition of auxin leads to recruitment of the proteasome and rapid degradation of your protein of interest. It looks wonderful on paper or on a screen like this. Most of the time, it doesn't work very well. We were very lucky that it worked extremely well for SPEN, and so within a short time of auxin induction, we could see very rapid depletion of SPEN, and that's shown here in this Western. So in the absence of tier 1, so which is essential for this auxin-degron system, you can see that even with the addition of auxin, you see no reduction in SPEN levels. But then in the presence of tier 1, when you add auxin, you can see that within an hour, SPEN has already disappeared. And what's wonderful about the system is that you can then bring back the protein by actually washing out the auxin in the cells, and the protein magically comes back because it's no longer being degraded. So this is really a very powerful system for looking at what a protein does immediately upon depletion, and also looking at what the consequences are once you've depleted the protein and then you bring it back. So we haven't actually explored all the possibilities that this system brought us, but I just want to show you what we were able to conclude with respect to what SPEN can do. And so here I'm showing you another one of these allelic maps by RNA sequencing. The allelic ratio is shown here. Here, this is not single cell. This is in the population, but we don't need to do single cell because we're inducing Xist using DOX. And so this is the control. Basically, without DOX, you can see biallelic expression of all these X-linked genes. Then when you add DOX, you can see within 24 hours, you've basically silenced the X chromosome almost entirely. Of course, the only gene that's still expressed from the silent X in blue is Xist itself, that you can see here. But then if you induce Xist with DOX, but at the same time you add auxin, you see that you get very little silencing. So basically, the presence of, the degradation of SPEN means that you abolish the onset of gene silencing. So François did this at various time points. I'm not going to show you all of the data, but indeed, the absence of SPEN leads to acute absence of gene silencing upon Xist coating. So we also validated this in vivo. As I said, we often go from ES cells to the mouse just to sort of have a reality check. And this is just to show you, we were able to look in previously published SPEN knockout mice. And what we did was actually use these mice, again, with polymorphic crosses. We could deplete both the maternal pool in the oocyte, but we could also deplete it zygotically. And so we could look at both the maternal knockout and the maternal and zygotic knockout and look at what the impact is on X inactivation, similarly to what I showed you earlier in the study by Maud. And this is a study that was done in collaboration with Jan Zilic, a postdoc in the lab, and in the mouse facility here at Curie. And so basically what we saw was that if you simply deplete the maternal pool of SPEN, you don't really see an effect. This is comparing the wild type to the zygotic, to the maternal depletion. But if you deplete both the maternal and the zygotic pool of SPEN, you basically see an absence of X-linked gene silencing that phenocopies the Xist knockout that I showed you earlier. So basically getting rid of SPEN is as if you got rid of Xist. So the SPEN knockout really is essential for X inactivation, and we see very similar defects at the blastocyst stage. So how does SPEN do this, and when does it come to the X? So this was something else that hadn't really been asked. There aren't very good antibodies to SPEN, unfortunately. Or at least not in our hands at the time. So what Francois did was take his SPEN, endogenous SPEN that was fused to GFP or to the halo tag, and we also knocked into Xist BGL stem loops that had been actually generated by Osamu Masuri, a previous postdoc in my lab, and therefore we could visualize Xist RNA at the same time as we could visualize SPEN in green. So again, this is still using the same inducible cell line that I showed you earlier, so we could switch on Xist and look at what happens both to Xist and to SPEN in living cells. So I'm not going to show you movies. I could have, but I don't have time to. But basically, this comes from the movies of SPEN and Xist visualization, and this was actually done by Julia Ranch, who was in the lab at the time, with the help of the imaging facility here at Curie. And basically what you can see is that as soon as Xist starts to accumulate, here in red, you can see that SPEN also accumulates. So the Xist and SPEN signals overlap almost entirely. In fact, the SPEN detection was slightly more sensitive than the Xist detection, so we could even start to see SPEN before we saw Xist. But this was just, we think, a detection effect. SPEN is not recruiting Xist is recruiting SPEN, and they really do come together at the same time. However, we know, and I'm not showing you this, that if you get rid of SPEN using the degron, you see no effect on Xist accumulation. So not having SPEN doesn't affect Xist coating and accumulation whatsoever. So we know that Xist brings in SPEN immediately, and so then the question was, well, what does it do? What does SPEN do? And which bits of SPEN are important? So what Francois did, and this was in collaboration with Agnès Lesseau, who was an engineer in my lab, they created a whole series of different constructs with which, where different parts of SPEN had been deleted, so that we could see what would rescue the SPEN phenotype. So using the line where we could degrade SPEN using the degron system, they knocked in these different fusion, not different fusion proteins, but different forms of SPEN where different domains had been deleted. The cDNA was knocked in to a ubiquitous locus in the genome, ROSA 26. So then SPEN, sort of different parts of SPEN were being expressed constitutively, and we could then ask the question: when you get rid of the endogenous SPEN by adding auxin, and when you induce Xist by adding dox, what happens? Well, so what happens is shown here. So this is the control. So basically here again, these are these allelic ratios. So basically, if you add Xist, if you induce Xist and you have SPEN present, you see gene silencing after 24 hours. And if you take away SPEN, you clearly see no gene silencing at all. If you add back full length, the full length degron, basically you see that gene silencing still happens, so you rescue completely. So this is what you'd expect, and we were reassured that the full length cDNA does rescue X inactivation in the absence of endogenous SPEN. And then by adding back these different bits of SPEN, so this is deleting one of the RNA binding motifs. This doesn't really seem to have much of an effect. You still rescue quite well. But if you delete all of these RNA binding motifs, here you do see an effect. And that's because SPEN is no longer being recruited to Xist, which is, so you'd expect that there would be an effect. But what's more important is that some of the conserved regions of SPEN were also tested, and this particular region, for example, doesn't seem to have an effect. The only region that seemed to have a dramatic effect was this Spock domain. If you delete the Spock domain of SPEN, you really don't see rescue of X inactivation. And so this is based on just a few genes here. What Francois did, of course, was an RNA-seq, and this seems to be a fairly global effect. So basically, the Spock domain of Xist seems to be the key domain. The Spock domain of SPEN is the key domain that seems to be involved in gene silencing on the X. So we know that SPEN is necessary and probably sufficient. To prove that it's sufficient, what Francois then did was an experiment that we all thought would be impossible to achieve, but he did it anyway, as a good student should. And so he took the Spock domain and tethered it to Xist. And so this is just the one little domain that we defined as being important for SPEN's function, we thought, although the other domains might still have a role. But without the RNA binding motifs, of course, SPEN wouldn't be able to bind. So he took just the Spock domain, and he tethered it via these BGL loops that I mentioned to you earlier to Xist, and looked then to see what happens with or without the SPEN degron. And basically what he found was that if you tether just the Spock domain to Xist, even in the absence of endogenous SPEN, you can rescue X inactivation of many X-linked genes. So this means that the Spock domain alone, recruited via Xist, is able to silence many genes. Now, it wasn't global. Some genes didn't seem to be as responsive as others, which I guess is reassuring. You would expect that you might need the rest of the SPEN protein to do something, but nevertheless, the vast majority of genes were affected when Spock was recruited to Xist. So we think, therefore, that the Spock domain is the key domain for X-linked gene silencing. So he also wanted to know, where does this protein go on the X chromosome to do what it does? Because it binds to Xist and seems to coat it globally, you would imagine that it would just carpet coat the whole X chromosome. That's what we were expecting. And so when you look at where SPEN goes, and I should say that to do this, what Francois did was use a technique, because we didn't have a good antibody for SPEN, and because CHIP with a bad antibody is no good, and because GFP CHIP wasn't great either, he went for another technique which came out a few years ago from Steve Hadnikoff's lab, which is called cut and run, that allows you to look at where a protein of interest binds. I don't have time to go into the details of this. It's been published. Many people use it. It's fantastically easy to use, and it's much cheaper than ChIP-seq. You have to have good controls, though. In our case, we had good controls because we were allele specific, and because we have the degron, we can say SPEN is here, SPEN isn't here, so it's actually easy for us to interpret the results of cut and run. So when you look at where SPEN binds using cut and run globally, what we found was when you induce Xist, you see no change in binding at autosomes. I guess you'd expect that. SPEN doesn't. Why should it change at autosomes when you induce Xist? But when you induce Xist and look at the X chromosome, you see that SPEN very rapidly increases in its binding on the X chromosome. But then, after a few hours, you see it actually decreases using cut and run. So this wasn't something that we'd really noticed by using the imaging, but we could definitely see it reproducibly using the cut and run. And when you look at the molecular level, and so here I'm not going to show you sort of huge maps of different regions. I'm just showing you one gene because it's really typical of what we see across the X chromosome. Basically, where SPEN binds, and so this is showing you the cut and run profile on the active and to be inactivated X, so blue and red here. So without Xist induction, you can see no SPEN binding really anywhere. But then when you induce Xist, you can see that SPEN starts to bind at the promoter of this X-linked gene. It goes up between 0 to 4 hours. It's still there at 8 hours, but it starts to go down at 24 hours. And this is what we saw globally across the X chromosome. Where we saw SPEN binding was mainly at promoters and enhancers of active genes. Inactive genes or silent genes, genes that just are not expressed in ES cells, didn't bind SPEN on the X chromosome, even though Xist was there. So we saw a few genes where indeed there was SPEN binding in the gene body. Those included Xist. Xist recruits SPEN, and perhaps as you'd expect, Xist actually is seen to harbor SPEN when Xist is induced. So basically SPEN does become enriched on the Xist locus itself. But in most cases, we only see SPEN at the enhancers and promoters of active genes. And then when the gene gets silenced, SPEN seems to decrease and to go away. So basically, we think that SPEN, through its Spock domain, actually initiates gene silencing via promoters and enhancers. So what Francois then went on to do, and here I'm actually skipping a lot because I do want to try and get a little bit in on the cancer aspect at the very end. But basically what Francois did, and this is in collaboration with the mass spec platform here at Curie, he took the Spock domain of SPEN and pulled out all the proteins that it interacts with in ES cells. And here is just, this is showing you the sort of families of proteins that he pulled out. So he pulled out basically NCOR SMART complex, which includes HDAC3, and this was something that we expected from previously published results, Oswald et al. It was also not a surprise to us because, as I'll show you in a minute, in a ChIP-seq analysis where we'd looked at histone marks, HDAC3 had already been implicated in X inactivation. So it pulls out this complex. It also pulled out the NERD complex, which was a bit of a surprise. It pulled out RNA splicing and localization proteins, which I guess one wouldn't be surprised by for what SPEN is supposed to do, although we haven't found a role yet for splicing in X inactivation, but there might be one. It pulled out the RNA methylation machinery, which had previously been shown to be recruited by Xist A-repeat. So in fact, we would suggest that it's the Spock domain of Xist that brings this machinery in to the Xist RNA and to the X chromosome. And for us, perhaps most satisfyingly, it pulls down POL2 and the transcription machinery. And given where SPEN seems to be located at enhancers and promoters, or active enhancers and promoters, this made absolute sense to us. So we think that indeed Xist recruits SPEN that brings down a whole panel of different factors via the Spock domain and associates it with POL2 and the transcriptional machinery to somehow inhibit transcription. And so this is a summary. The paper just came out literally yesterday, I think. So it's now online if you want to read it. So basically, this is a model of what we think is happening. So Xist, when it becomes upregulated, produces an RNA that SPEN will bind to via the A-repeat. Through the Spock domain, this actually brings in a whole host of different factors, including the RNA modification machinery. But it also means that SPEN, through the Spock domain, links up to chromatin on the X, that links up to enhancers and promoters. And this is where we think the silencing effect happens. And what Francois is doing right now is using structural biology approaches to work out how Spock interferes with transcription. And so that will be hopefully something that we can talk about in a few months' time. But basically, we think that Spock does interfere with the process of transcription. But once silencing is achieved, although the Xist RNA is still there and SPEN is still there, it no longer binds to X-linked genes. It disengages. So basically, although it's still being brought in, it will not engage with chromatin because that doesn't have active transcription and therefore polymerase, and presumably also these factors such as NCOR SMART and HDAC3. So it wasn't at all what we expected, but this is what we found. And I think that this is one of the first sort of molecular hints as to how gene silencing really works during X inactivation. But I just want to point out that although Xist is important for initiation, it still coats the chromosome even after initiation has happened. It still brings in factors, including SPEN. So we actually looked in differentiated cells, such as neural progenitor cells, to see whether SPEN is still there, and it is. You can see this is Xist RNA fish. This is SPEN detected using the halo tag that I mentioned earlier. So indeed, when we look at differentiated neural progenitor cells from this system that I already showed you, we can see that SPEN is there. So what happens if you deplete SPEN in this system, where you have an inactive X, where genes are silent, there's already DNA methylation? What happens? Well, when you deplete SPEN, either 24 hours or 48 hours, you see very little effect on X-linked gene expression, which means that you have no effect really on the silent state. So SPEN is really involved in the initiation of X inactivation. Once you've shut down the genes completely, SPEN no longer goes there, as the RNA, as the cut and run showed, and B, it doesn't really seem to have an effect. But where it does have an effect is in a handful of genes that escape X inactivation. And that's shown here. We see that indeed, when you deplete SPEN, a few genes do seem to show an upregulation, and these are these X-linked escapees that I mentioned to you earlier. And so basically, this means that SPEN, even in a differentiated cell population, does have an effect on genes that are expressed on the X. These genes somehow avoid the whole process, and yet they are slightly susceptible to SPEN. And when you get rid of SPEN, you can see that their expression goes up. So does this upregulation matter? Well, in NPCs, it doesn't seem to, because they don't die, but we haven't looked in vivo. And now, if I have time, and I don't know whether I've run out of time, I've forgot. Okay, good. And now if I have time, I'm going to talk to you a little bit more about what we think might happen in other situations where Xist is compromised in a situation where the X is supposedly fully inactive and yet Xist is compromised. Anyway, so basically SPEN is important for initiation. It's not so important for maintenance, but it does affect escape. So actually, I want to say just a brief word then about the chromatin changes, because I've shown you, I hope I've convinced you, that one of the key factors for X inactivation through Xist is SPEN. It does seem to have a role via HDAC3. We'd already actually started to look at what happens at the chromatin changes during X inactivation in a cell in cells using both RNA fish, as you can see here. So this is Xist RNA and H3K27 trimethylation. So we knew that these marks come on quite early, but we never really looked at the timing, the high resolution timing during initiation, and that's something that we did, and this was published last year, so I'm actually not going to go through it. I have a few slides, but I'm actually going to skip them because I do want to get to the cancer part. But basically in this paper what we showed was that using the same cell line, if you induce Xist, what are the different marks that come on during X inactivation, the histone active marks, the histone inactive marks, such as polycomb-associated marks. So we use the same system, non-random X inactivation. We use native ChIP-seq in this case, and we also used RNA-seq and TT-seq to look at TT-seq, which is another form of RNA-seq, but more sensitive, to look at gene silencing. And all of this was allele-specific. And I should say it was an epic effort by Jan Zilic, a postdoc in the lab, who's actually just about to start his own group in Copenhagen, and Aurélie Bouzard, who was the bioinformatics postdoc in the lab, who's actually now moved to Brussels. So they were a dream team who managed to get this epic amount of work together. And I should say that this was only possible because it's very expensive, thanks to an ELC. Thank you, ELC. So we looked at a vast range of different marks. I'm not going to go through all of them. I just want to show you that histone deacetylation seemed to be one of the very earliest marks during X inactivation. And this is showing you both at the global level, but allele specifically, what happens within 4 to 8 hours of Xist induction. You can see that on the black 6, which is the Xist-coated chromosome, you see reduced levels of H3K27 acetylation. So loss of K27 acetylation is faster than any of the other marks we looked at, including H3K4 monomethylation, which is actually known to be associated with enhancer marks. So this was a very rapid loss compared to the other marks, which led us to indeed look at what this might be due to. And we tested all of the histone deacetylases to find if they were important. So I've kind of already given away the result, but I should say that in this paper we tested every single HDAC to find out whether they were involved in X inactivation. The only one that showed an effect when you either knocked it down or used a chemical inhibitor or knocked it out, and we did a knockout in those cells, was HDAC3. So HDAC3 really does seem to be involved in promoting efficient gene silencing. And so we looked at this at the chromosome-wide level in these knockout cells, and we found that indeed in this particular gene, you can see in a wild-type situation, you can see the levels of H3K27 acetylation go down on the inactive X, whereas in the HDAC3 knockout situation, the levels haven't changed. So clearly HDAC3 is important, but it turns out that many genes on the X seem to not be affected by this, which suggests that what SPEN via its spot domain is doing is not just via HDAC3. There clearly are other mechanisms for gene silencing that could involve chromatin. But this for us was actually quite important because it shows that chromatin does matter for the beginning of gene silencing. It's not just a consequence of the silent state. So this is just what I was saying, that there are some genes that seem to be less sensitive to HDAC3 loss, and the question is, how are these regulated? So we also looked at the polycomb-associated marks, and here I'm showing you H2A ubiquitination and H3K27 trimethylation. And what we found was that H2A ubiquitination seems to be a very rapidly induced mark on the inactive X, much faster than we imagined actually, and actually faster than H3K27 trimethylation. So this was a big surprise to us, although perhaps not to others in the field, because Neil Brockdorff and others had already predicted that PLC1, which is what the complex that brings down H2A ubiquitination, probably precedes PLC2. This is contrary to what others have published in the field. In particular, the Lee Lab have proposed that PLC2 is the very first mark that's brought in by Xist. In fact, it's probably PLC1 that is recruited by Xist, or some of its cofactors, via HNRMPK, and that was shown by Neil Brokdal's lab. And this actually fits very well with that conclusion, which is that indeed PLC1 comes in very quickly, not via the A-repeat region, via another part of Xist. And this is what actually then leads to the accumulation of PLC1 and then PLC2. However, and this is something that I think is important for everyone to bear in mind, although polycomb is recruited by Xist very quickly, these marks will only spread into genes once they're silenced. So you need the Xist A-repeat, and you need SPEN, and you need gene silencing in order for these marks to actually spread into the gene bodies. And this was shown actually by Harel again, and this was actually a study that was driven by Simao, a postdoc in my lab, who has now set up his own lab. So this summarizes what we saw and what others have seen as well. Basically, we think that indeed the very first event that happens during Xist-mediated silencing is SPEN, but also a loss of active H3K27 acetylation. You also see an increase in H2A ubiquitination in non-genic regions that can spread into genic regions once gene silencing has actually happened. And H2A ubiquitination is the first mark. H3K27 trimethylation then follows and comes in slightly later. So this is indeed the roadmap of initiation of X inactivation at the level of chromatin. And this is a cartoon just to show you how we think all this happens. We think the A-repeat of Xist is bringing in SPEN. This probably somehow activates and helps to bring in HDAC3 at enhancers, mainly. At promoters, it's probably doing something else. And the other parts of Xist, and notably the B region of Xist, probably bring in HNRMPK, which brings in non-canonical PLC1, and this leads to ubiquitination of histones, which will then lead to recruitment of PLC2, and then spread of these marks. So we think that this order of events means that you progressively lock in X inactivation, because we know that these polycomb marks are involved in the maintenance of silencing, because if you take them away, you do see a loss of gene silencing. So these are the conclusions of the first part of my talk, thinking that I wasn't going to get to the second part at all. Can I zip through in 5 minutes? Okay, really, 5 minutes, I promise. Okay, so I'll come back to the conclusions for those of you who are interested. I hope I was clear enough, you know what's going on. So one of the main interests, okay, I really will come back to this. If those of you who are really interested in X inactivation, you can take a photo of this, or you can watch it, or I can give it to you, or you can just read the papers as well. Anyway, we could talk about it, but what really interests us in the lab right now is also escape, and how do you escape from X inactivation? How often do genes escape? What are the mechanisms? Does it matter? And does it happen in cancer? I already showed you, what's fascinating about escape. We looked at the chromatin level, what happens using all these different maps that we generated. What do you see at a gene that escapes? This is a constitutive escapee. What do you see? Well, nothing very interesting. You don't see a loss of active marks, and you don't see a gain of the inactive marks, and that's shown here. This is K27, this is H2. So you don't see really much change. Well, so what? That's kind of what you'd expect. That still doesn't tell us how escape really happens. We know that SPEN matters, though, for this dampening, but it's not clearly managing to shut down genes completely. So one of the hypotheses that we've come up with based on looking at chromosome organization is that the way these regions are organized could be linked to their capacity to escape. So this also has been published, so I won't go through it in detail, but genes that escape, at least facultative escapees, they tend to escape in clusters, and they seem to form topologically associating domains on the otherwise tadless inactive X. And so we think that this could be somehow linked to their capacity to escape, and this is something that we're trying to work out. I won't go into the details of it, but this is something that Agnese Loda, a postdoc in the lab, and Antonio Houth, a student in the lab, are trying to work out what is the relationship between X-linked gene escape and 3D chromosome organization. But what happens in cancer, where no one had actually really looked up until a few years ago? We started to look at cancer cells here at the Curie Institute for obvious reasons. It had been actually shown by Barr many, many years ago that in cancer cells, contrary to normal cells, where he picked up what's known as what still is known as the Barr body, the inactive X in a normal cell. This seemed to be lost or impossible to identify in cancer cells, where massive nuclear changes seem to happen at the chromatin level. So it was already reported that the sex chromatin, as it was called then, before we knew it was the inactive X, was somehow lost in malignant tissues. And what Ronan Chalinier did in my lab several years ago, in collaboration with Anne-Vincent Salomon and Marc-Henri Stern and others here at the Curie Institute, was take a look at what happens in tumor cell lines, breast cancer cell lines, but also in primary tumors. And this is showing you what a normal cell looks like at the DAPI level. You can see the inactive X beautifully here. Xist RNA is coating it. And this is in a basal-like breast cancer cell. You can see that this is Xist coating. It seems to be all over the shot, and you really don't see a dense body anymore. And so we looked more carefully, and this was published, as I said, was conducted and published a few years ago in Genome Research. So we looked both in cell lines and in primary tumors, and we found that indeed you do often see, well, first of all, you see multiple X's, sometimes inactive, sometimes not. You see Xist RNA is aberrantly coating the X. It seems to be more lowly expressed often. We see global chromatin disorganisation using ChIP-seq, but also using IF. And we did find that there are X-linked genes that become re-expressed. So X-linked gene RNA FISH showed that there are genes that seem to come back on in the context of cancer. And so this is something that we proposed might have an impact on tumor growth or progression, and it's something that still needs to be tested. But I wanted to very quickly end on two slides that have tried to explore this to some extent and that came at it from a slightly different angle. And I hope that the authors of this study that hasn't yet been published are here. I think I can see Christophe up there. Hopefully Raphael is here. No, he's not here. But Laia, maybe? Laia's here? Yes? No? Yes? Ah, good. Okay, perfect. So I'm going to just say two words about this, and then if there is interest and discussion, I'm glad they're here because they can present it even better than I can. So basically, what we showed in the breast tumors and the cell lines that we looked at was that there is erosion, and so this is what this shows: an eroded X, inactive X, where Xist is still expressed. But you do see some gene expression, and so the red squiggles are actually X-linked gene expression. And here you can see that although some gene silencing is there, you do see some gene expression as well. So there is some transcriptional reactivation, and we reported that this can lead to changes in gene dosage at the RNA level, maybe at the protein level, but not something that we'd really explored very much. So very much still an open question. Does it have an impact? And if it does it matter? And so this is a study that was then taken on by Laia, who actually was a co-postdoc between my lab and Raphael Margrand's lab, but mainly Raphael's lab. And this was a study that was done in close collaboration with Christophe, who's up here, and his and student Marie-Lou Picot, and his collaborators in Marseille, where they had already published a study or shown that indeed losing Xist can compromise the capacity of mammary stem cells to differentiate. And so in collaboration, we've been looking, or they've been looking, to see does loss of Xist actually lead to a problem in X-linked gene dosage and lead to any defects? And this summarizes the results so far. So basically, indeed, if you knock out Xist or if you silence Xist using a CRISPR dead-Cas9 approach, you see that actually this leads to a block in the stem cell compartment. And this is using human mammary stem cells here. I'm not talking about the mouse. And so you can see that in a wild-type situation, these cells go through differentiation. But when you knock out Xist or you block it, you see that most of them actually remain in this stem cell compartment. And what happens, and this is where Laia came in to actually look at what goes wrong using RNA-seq and then also ChIP-seq, etc. And what she found was that actually there is a subset of X-linked genes that comes back on. This parallels very well what we had been seeing in the breast cancer situations. And what she went on to show is that these genes actually seem to cluster and they're found in proximity to SKPs. And when she zoomed in and looked at what else seemed to correlate with them, it seems that they are linked to PLC1. And Xist, we know, can recruit PLC1, so this could be part of what goes on. When you lose Xist, you no longer have PLC1, but you also no longer have SPEN, I should remind you. And what she found was with one of these genes, which was a key gene, MED14, so this is one of the critical subunits of the mediator complex, which I'm sure many of you will know is involved in transcriptional regulation and enhancers. What she found was that in the situation where you knock out or repress Xist, you see an increase in the expression of MED14. This is an X-linked gene that shows partial escape. It comes on more fully, and this leads to higher protein levels of other mediator subunits, and this leads to a global increase in enhancer activity. And so this suggests that indeed the loss or downregulation of Xist in some cancer cells, or the delocalization of Xist in some cancer cells, might be linked to what happens and this could be mediated by some of these genes that become transcriptionally de-repressed, or escapees that become upregulated, which could then have genome-wide effects. So this is a work in progress. It's ongoing. I won't say more. If you do want to ask questions, you're welcome to. But I think this shows you that indeed, despite the fact that we all thought Xist was just involved in the initiation of X inactivation early in development, and that was it, clearly there's a whole universe that we do now need to explore in the context of stem cells and in the context of cancer. And with that, I will end. I will not tell you that dosage matters. I hope I've convinced you. And I will just acknowledge the people who did the work. So this is my lab after we moved to Heidelberg. In fact, this is not Heidelberg. Those of you who know Heidelberg know that it's not so sunny. This is actually a retreat we did in Barcelona. I hope I've mentioned all the names of the people who did the work. The people with the yellow halos are the angels who followed me to EMBL Heidelberg. And I want to thank all of them and everyone in my lab who I've been so lucky over the years. It's amazing the people I've managed to work with. And I do want to acknowledge our collaborators, in particular the Curie Institute that hosted me for so many years. We did such fantastic things here in collaboration with people, in particular the imaging team and the bioinformatics I didn't mention, but Nicolas Servon and Emmanuel Barriot have been long-term collaborators of ours and continue. Also, the animal facility, which I mentioned, was key to some of the work that we've done. And our outside collaborators, this is just a handful of them. Forgive me if I've forgotten some of the people I mentioned here, but hopefully you all got mentioned. But so Thomas, who actually shared the SPEN mice with us, Alina, who I mentioned, Raphael, and many, many others. And I want to say that although the move to EMBL has been extremely exciting and a wonderful adventure for me and my team, it's also been quite painful, and I really do miss this institute. But, don't tell EMBL, please. And, of course, the Collège de France, who've always been there in a sort of wise way to guide me through all that I do. So thank you for your attention, and happy to take questions. And I'll go back to my conclusion slide. That way, in case anyone was really interested, you can read them. Thank you.